Everybody, thanks for joining online. We are once again doing an in-person medical grand rounds. We're going to keep trying to do these the first grand rounds of every month, and we expect to see. And I'm already starting to see more people come than last time, so that's a good trend. And we'll hopefully continue to see more and more people showing up. I see my ward team is here today. Thank you guys for coming. So we have a really amazing medical grand rounds by Dr. Bhatt today. Before we do that, I just want to turn over to Dr. Salas, who's going to have a few updates for us. Okay.
Thank you so much, Errol. It's great to see everybody this morning. And in honor of Native American Heritage Month, we wanted to share with everyone the university's new, as of last month, land acknowledgement, which you can find online. I have the link on the next slide. But I just wanted to read it for us all this morning. Stanford sits on the ancestral land of the Muwekwa Ohlone tribe. The land was and continues to be of great importance to the Ohlone people. Consistent with our values of community and inclusion, we have a responsibility to acknowledge, honor, and make visible the university's relationship to Native peoples. Sorry, how do I advance? This one, oh, sorry. For folks on the Zoom, there's 3 screens here, so I wasn't sure which one. If you want to learn more about the Stanford Land Acknowledgement, that QR code on the left will take you to a website that has a video, including some of our Stanford students and some more information on the history of Stanford and Indigenous peoples. And on the right, I wanted to include the QR code for the Stanford Native American Cultural Center, which has also a listing of really excellent events that go on throughout the entire year. And we wanted to just reinforce that while land acknowledgements are important, and that's why we're doing one today, they're not sufficient really for counteracting all the harm that has been done to Indigenous communities. So we really encourage everyone to think about and commit to taking some actions to support Indigenous communities, either by getting involved in grassroots change movements or donating or otherwise supporting organizations that are doing that work. And I would say the Native American Cultural Center is one of those, and there is a link on their website to donate if you would like to do so. Also, we will be holding an inclusion rounds. For those of you who've been following our diversity-related events, we've been having inclusion rounds every 1 to 2 months about different topics, and we are going to have one about Native American Heritage Month, so keep an eye out. We will send out information, including the speakers and the time and date. And lastly, we also want to acknowledge that tomorrow is Diwali, so happy Diwali to everyone who celebrates, and I'm going to pass it back over to Errol.
Dr. Salas, also thank you to Drs. Dunn and Harmon for always making sure that we are celebrating and honoring all the important events throughout the year, and we'll continue to do so, as well as please keep us updated if we are making sure that we're honoring all the right things. So please let us know if there's anything that you'd like to see us be doing more of. It's really important that we make sure everybody feel part of this community as much as possible. So please let us know. Thank you again, Dr. Salas. And now I'm excited. one thing before I introduce our speaker today. Next week, we are celebrating 50 years of Stanford nephrology. Actually, this week is National Kidney Week, and there's a lot of meetings going on for the American Association of Nephrology. So we'll have Drs. Chang and Jameson talking next week, and then we'll have you updated on the following week. We'll, of course, towards the end of this month for Thanksgiving, now have grand rounds during that week as well. But now I'm excited to introduce my friend and colleague, Dr. Ami Batt. Dr. Ami Batt is Associate Professor of Medicine and Genetics at Stanford University. She completed residency and chief residency in internal medicine at Brigham and Women's Hospital and was a fellow of hematology and oncology at Dana-Farber Cancer Institute. Thereafter, she carried out her postdoctoral duties at the Broad Institute of Harvard and MIT. Her lab seeks to improve outcomes in patients in hematological malignancies by exhaustively characterizing the dynamics of the microbiome in immunocompromised individuals, and exploring how changes in the microbiome are associated with idiopathic diseases in the population. She loves working with trainees, and she's always excited about the application of new molecular and computational technologies to solve complicated metagenomic puzzles. She works, and looking at her lab, she has grad students, medical students, undergrad students. She even has a program for undergraduate and high school students who are interested in the summer research initiatives as well. We don't have enough time to go through all the honors awards, but I'll mention a few of the most recent ones. She's elected member of the American Society of Clinical Investigation. She's a National Academy of Medicine member as well as of 2020, Sloan Foundation Fellow from 2020, Chen Award in Excellence for the Human Genome Organization in 2018, and literally like the whole a list of awards and awards just over the recent years. Ami is committed she's committed to improving cancer care and education research in resource-limited settings. She's the director of global oncology for the Center of Innovation and Global Health at Stanford. She's a co-founder and president of a non-profit organization called Global Oncology. And this is an amazing organization. And knowing Ami, I've gotten to learn more about this over this past year. And 80% of cancer deaths in the low to middle income countries, so 80% of cancer deaths, despite getting only 5% of global funding. And so they're doing all sorts of amazing programs. So 80% compared to AIDS, tuberculosis, and malaria combined, there's way more cancer deaths, and they're just not getting funded the same. So her job is to make sure they're getting the funding, attention, and really educating people in various countries so that they are getting this higher level of care to protect from cancers. One example is the cervical cancer-free Nigeria campaign, and the former president of Nigeria is the ambassador for that. So it's not just, this is a huge program she founded and is part of. It's really an amazing work that she's doing, in addition to all the other amazing things that she does. And on top of that, she was also active with this past couple of years in regards to COVID. And she's actually here to talk to us about with the title, The Ghost of SARS-CoV-2, a large-scale analysis of fecal shedding in mild COVID. So Dr. Bhatt, thank you so much for being with us today. And we have more and more people coming in as well. So we're getting more in person as well and lots online. Thanks for being with us.
All right, thank you very much. Thank you, Errol, my friend and colleague, for that amazing introduction. I hope I live up to the hype, but at least I'll tell you some good jokes. So I think it's very fitting that I get to give the #2 in-person grand rounds. And today I'm going to be talking about the ghost of SARS-CoV-2, a large-scale analysis of fecal shedding in mild to moderate COVID. These are my disclosures. The majority of these are not relevant, but I will call your attention to the bottom one. We do have a patent filed, a provisional patent filed on the SARS-CoV-2 RNA detection method that I'm going to be presenting. And this is the very most important slide of my presentation. This is a picture of my absolutely amazing research group. I am privileged to work with some of the smartest young people in the world, and they are not only brilliant and fun, but they are committed to trying to make the world a better place. So I thank my lucky stars every single day for having colleagues like this. So in our lab, we think a lot about the gut microbiome. This is a beautiful cross-sectional image of the colon of a mouse. But our colons look pretty similar. This picture was actually taken by Kristen Earle, a former graduate student here at Stanford, when she was in the Sonnenberg lab. In the bottom right-hand corner, you can see the DAPI counterstain nuclei of the epithelial layer of the colon. That's the blue part. In the top left-hand corner, you can see a variety of different bacteria that are filling the lumen of the gut. And between the two, you see these green layers. You'll notice that there are two different layers. One is the thick layer of mucus, and the other is the thin layer of mucus. As a lab, we've spent several years exhaustively trying to characterize the microbiome. I think of us as measurement technologists. We develop methods to measure the microbiome better, with the hope that precision medicine will be derived from these precise microbial measurements. We've answered detailed questions like who is there, and not just at the genus or species level, but at the strain level. And more recently, we've been digging into trying to figure out what these microbes are doing, what they make, and how they talk to one another. In our day-to-day world, we like to zoom in to this area. And this is what we envision is happening. There are millions of microbes that live there, in fact, trillions of thousands of different varieties. And we anticipate that they are making molecules that signal to one another and to us. Because of this, over the course of the past several years, we have not only made tools to study the gut microbiome in humans, especially as it relates to human health, but we've also become really, really good at collecting fecal samples. Now, I know pride is not something to be, pursued, but I am actually very proud of our freezers full of stool. And when COVID hit, like many of you, we were trying to figure out how we could make a contribution. So we've had the chance to collaborate extensively with a variety of different investigators all over Stanford. And what I'm going to present, which follows, is really the collaborative work of a lot of people listed here and a lot of people not listed here. I'd like to particularly call attention to Press Jagannathan and Upi Singh, who led the lambda interferon clinical trial from which we collected samples, Haley Hedlund and Alex Dahlin from the Quantitative Sciences Unit, and Arvind Natarajan, Sumaya Zlitny, Aaron Brooks, Summer Vance, Alvin Hahn, Ryan Park, and Danica Schmidtke from my lab, who have contributed extensively to this project and other related projects. So when COVID-19 hit, we decided that we were going to do what we were best at, which was collecting stool. And you might ask, why collect stool in COVID-19? It's a respiratory disease as we think of it. But studies had shown at that time, even early on, that COVID-19 patients can shed SARS-CoV-2 RNA in their stool for up to a week after symptoms subside, even after nasal swabs tested negative. And so our question was, could the virus be infecting the GI tract? And furthermore, could the presence of SARS-CoV-2 in the feces be associated with clinical symptoms, predictive of outcomes, or useful for other purposes, such as epidemiology? We hoped that by studying stool samples from patients with COVID-19, we could answer some of the following questions, including, is there evidence for potential viral infection in the gut? How long is viral RNA shed in the stool? And how does the timeline of shedding in the stool compare to the course of active disease? Collection methods are out there to collect stool samples for the purpose of microbiome studies, and we leverage these heavily to try and collect stool from patients in the outpatient setting. Stool is pretty non-invasive and easy to collect, and we thought that it would be a good proxy to show us what is happening in the digestive tract. And so we created a flyer. This is actually images that were made by Ryan Brewster, a former medical student here at Stanford from our lab. We created this flyer advertising our amazing effort to collect stool. Now, the reasoning behind this from a scientific perspective was that GI symptoms were emerging as common in COVID-19. For many of you who practice clinical medicine, around that time, it was kind of fascinating to note that not only did people have respiratory symptoms, anosmia, but it was emerging that a subset of patients had pretty substantial abdominal pain, diarrhea. And in fact, once testing really got up and running, we noticed that a lot of people had GI symptoms in the absence of some of the more standard symptoms of COVID-19. And so beyond just respiratory symptoms and systemic symptoms, GI symptoms have emerged as relatively prevalent in this patient population. At this point, well into the pandemic, we have lots of analyses and meta-analyses that are in the literature. And the suggestion is that GI symptoms occur in about 20% of subjects. Over the course of the same time, a lot of research has been done to try and ask the important question of, can SARS-CoV-2 infect the GI tract? And lest you think that this is kind of a strange question to be asking, I'll remind you all that coronaviruses are known to infect mammals. They can infect mammalian guts, and indeed feline coronavirus, for example, is a common infection of cat guts. There's even a vaccine against the feline coronavirus. So the idea that a coronavirus can infect the GI tract is not outlandish. As I mentioned, over the course of the past year and a half, a lot of research has emerged to suggest that there might be a patent infection of the GI tract. We know that the receptors that are important for SARS-CoV-2 binding and internalization are expressed on intestinal epithelia, such as the ACE2 receptor and TMPRSS2, which may be important for cleaving certain coronavirus proteins. We know that fecal calprotectin is actually elevated. This is a marker of inflammation, and this was beautiful work that came out of Stanford's GI group. We also know that there is IgA, or a weak signal of an immunoglobulin response, a secreted immunoglobulin response in the stool to an RBD protein. There have been measured elevations of IL-8 and IL-23. And of course, to this point, there had been measurements of genomic RNA of SARS-CoV-2 in fecal samples. In very impressive work that came out of Hans Cleaver's group, it was demonstrated that SARS-CoV-2 can not only bind to and infect colon epithelial cells in vitro, but it can also actually bind to and infect organoids from the intestine. And here are images of the duodenal organoids, ileum organoids, and colon organoids from these types of studies. The question of whether or not the GI tract can really be infected and if the GI tract is a source of infectious particles is far from settled. There's in fact a continued raging debate on this topic, although there have been limited reports of scientists being able to isolate live virus from fecal samples and use this virus to infect vero cells in vitro. Now, of course, as I've mentioned, these are kind of few and far between in terms of reports, and so it's very difficult to know for us at this point whether or not SARS-CoV-2 can be spread through fecal-oral methods. But we do know that SARS-CoV-2 RNA can be detected in fecal samples. We also know not only that the RNA can be detected in fecal samples, but that SARS-CoV-2 nucleocapsid protein can be detected in the GI tract, surprisingly up to months after infection. If we're thinking about the potential of SARS-CoV-2 to infect the respiratory tract and maybe make it down the esophagus and be detected in the fecal samples, it would be surprising if nucleocapsid protein that was kind of being sloughed out of the respiratory system and then back down the esophagus was present in these actual gut samples so long after respiratory infections are cleared. And so this has led us all to the question of whether or not there might be long-term low-level infections in the gut, and whether or not this might be related to long-term immune effects. We became fascinated by this as more and more papers were coming out, and I had the opportunity to write a perspective with Aaron Brooks, a staff scientist in our lab, where we not only investigated the literature on SARS-CoV-2 infections in the gut, but also the potential role of the gut microbiome in modulating this infection and its symptoms. What we really did, though, is, like I told you, what we were great at, which was figure out a way to collect stool samples from these patients and study them. And so this is a picture of a Zoom call between our COVID team. Aaron Brooks, Alvin Hahn, Sumaya Zlitny, Summer Vance, and Aaron Natarajan are pictured here. They have really formed the core of this team. They've worked tirelessly through very difficult circumstances to bring this research to you, and I'm very happy to be able to share both published and unpublished data with you next. So before we jump into it, I would argue that robust detection of fecal SARS-CoV-2 RNA is valuable to us. It might help us understand the course of viral shedding vis-a-vis patient health. This may help us understand what the consequences are of extended shedding, if any, and if extended shedding might be related to an immune response or a lack of an immune response. There's obviously epidemiological relevance to this. I think by this time, all of us are aware that wastewater-based epidemiology has been used extensively, both here at Stanford and worldwide, to do community surveillance. But in order to take those data from wastewater surveillance and turn them into real estimates of the percentage of populations that are actually infected, we need to have a clear idea of how much SARS-CoV-2 RNA is being shed and over what time course. I would also argue that to be able to assess this properly, we need this information not only for patients with severe COVID who are in the ICU, but also the vast majority of patients with COVID who are actually in the outpatient setting. Now, there also might be clinical relevance to these findings. Stool is obviously an additional monitoring opportunity for SARS-CoV-2, and it might be related to symptoms, which could be fascinating. Unfortunately, when we started this project, methods to measure fecal SARS-CoV-2 RNA were not benchmarked, highly heterogeneous, and used inefficient strategies, which meant that it was very difficult to assess the sensitivity and specificity of these approaches, and it was very difficult to compare between studies. And so at this point, what we really needed was a measurement method that we could trust. And I'll remind you that as a lab, we've been very focused on that idea of how do we measure things properly. So our first effort, while we were collecting samples from clinical studies, was to develop methods to collect stool, preserve fecal SARS-CoV-2 RNA, and then detect it. This is work that was led by Arvind Natarajan and Alvin Hahn. In our first effort, we added synthetic SARS-CoV-2 RNA, as well as a positive control, an attenuated bovine coronavirus, to either NIST the National Institutes of Standards and Technology's standardized stool or healthy stool from healthy individuals. We then preserved these samples in 3 different ways: in the classically used OmniGut DNA genotek tube, which is commonly used in microbiome studies for DNA-based applications, the Zymo DNA RNA shield tube, and finally, without a preservative in just phosphate buffered saline. We then tested 3 different extraction kits: the MagMax, the QIAamp, and the Zymoviral kit. And then we detected SARS-CoV-2 RNA using 2 different methods, both digital droplet PCR and RT-qPCR. What we found was that in the case of the NIST standard stool, the ZY preservative worked best and the Zymoviral extraction kit worked best. We found that ddPCR and RT-qPCR were fairly comparable. And I'll just note that given the high expense of ddPCR compared to RT-qPCR, this really suggested that RT-qPCR might be a preferred method in terms of generalizability. With the healthy stool, we found that the ZY preservative again outperformed the OmniGut preservative or PBS. In this case, however, we found that the QIAamp extraction kit performed better, and we did find that ddPCR slightly edged out RT-qPCR in terms of sensitivity, but was roughly comparable. So we then decided to test this on real patient samples. And we were very fortunate to get samples not only from the Lambda study, but also from inpatients, thanks to Angela Rogers and Catherine Blish. We were able to take these stool samples, some of which were preserved only in Zymo, some of which were preserved in both OmniGut and Zymo. We extracted with a variety of different methods, and we found that more detectable RNA was present with the Zymo preservative kit using the QIAamp extraction kit. And so in conclusion, we decided that the Zymo preservative works best and the QIAamp extraction performs the best. Now, I appreciate that this is kind of technical and perhaps a little boring, but if we're really trying to measure things properly, it's critical that we have methods that are reproducible and rigorous. And for that reason, we pulled together all of these experiments in one of the most extensive benchmark studies of how to detect fecal RNA in stool, and we published this manuscript. So I mentioned that we've collaborated extensively with clinical collaborators who are testing various interventions to try and identify therapies that might modulate COVID severity or duration. One of the very first of the studies to come out of Stanford was a study of interferon lambda. Interferon lambda is an immunomodulatory agent that has shown some promise in the treatment of certain hepatitides, and it is administered as a single subcutaneous injection. In this trial, patients with mild to moderate COVID were enrolled at the time of diagnosis, and they were randomized 1-to-1 to either receive peg interferon lambda or to receive a placebo control. The goal was to test whether or not lambda could affect coronavirus shedding or symptoms. Now, one of the big advantages of this clinical study was that it was very carefully designed to not only test its primary endpoint, but also to do extensive sampling of blood, oropharyngeal swabs, clinical evaluation, anterior nares swabs that were self-administered. Patients filled out longitudinal symptom surveys, and we also collected longitudinal stool samples out to 10 months after the original infection. This is actually what the sampling scheme looks like. It's complicated, and one thing that is absolutely amazing is that the compliance with this study was outstanding, which means that we have a very complete set of not only samples, but symptom data as well, and symptoms that were collected in a routine manner. One of the other big advantages for us being very interested in whether fecal SARS-CoV-2 shedding was associated with GI symptoms is that this is one of the few prospective studies that took great care to collect information about GI symptoms, not only respiratory symptoms, which we thought were more common in SARS-CoV-2. So while the actual Lambda trial ended up being negative, there was no advantage to Lambda versus control, this formed the basis of an outstanding sample collection with which to study the dynamics of fecal shedding in this patient population, and also the relationship between fecal shedding and symptoms. To give you a sense of our cohort characteristics, we were about equal, a little bit unequal in terms of males versus females, with 70 males enrolled and 50 females enrolled. You can see that the age ranged from 18 to 71. And we had a fairly similar age distribution across sex at birth. When we compared laboratory characteristics of individuals stratified by GI symptoms at baseline, so you'll see that we have people who had GI symptoms at baseline and people who did not have GI symptoms at baseline, we did not find any substantial or notable differences in baseline laboratory values, including white blood cell count, absolute lymphocyte count, and liver function tests. I'll also note that the baseline seropositivity, meaning the presence of antibodies at the initiation of the study, these are antibodies to SARS-CoV-2, was not different between the two studies. When we take a look at clinical characteristics, similarly, we don't find that, for example, women are more likely to have GI symptoms than men. We also didn't find any notable differences in terms of GI symptoms segregating by race and ethnicity, BMI, or duration of symptoms prior to randomization. Although I will note that we're underpowered to actually do a lot of these comparisons. This is just to give you a sense of our cohort. So the first thing that we found applying these methods was that long-term fecal shedding was observed up to 7 months after enrollment. In both of these graphs, fecal shedding is indicated in the brown bar, and oropharyngeal shedding of SARS-CoV-2 is indicated in the gray bar. We have the percent of positive subjects on the y-axis in both graphs and time point indicated on the x-axis. Over on the left, you can see the shedding that happened in the early time points after enrollment, so in the first 28 days. And I'll call your attention to the fact that at the earliest time point, we saw more patients having oropharyngeal shedding than fecal shedding, but over the course of time, that relationship inverted. And if you take a look at the panel B, which is on your right side, what you'll find is by day 120 and day 210, we have no patients in whom we can detect respiratory shedding, but we have many patients, or actually a good number of patients, I should say, in which we can continue to detect fecal shedding. So up to this point, the longest shedding that had been reported was about 70 days. And you can see that we're going out to 4 months and 7 months here. Now, one thing I don't know the answer to is whether or not these could be the consequence of reinfection, or if these were the same strains of coronavirus, although we do have an ongoing collaboration with Illumina to try and sequence the full SARS-CoV-2 genome from these fecal samples. And we have been able to sequence the full genome of SARS-CoV-2 samples in individuals who have a high fecal titer. Now, we not only measured these with one target RNA, we actually looked at a variety of different target RNAs of SARS-CoV-2. Here on this stack bar histogram, you can see time on the x-axis, percent of subjects testing positive on the y-axis. Blue indicates no RNA was detected, and the different colors indicate differing amounts of viral RNA being detected, with the light blue being the very highest amount of viral RNA. What you can see is that the findings for the 4 genes tested here, the E gene, the N1 gene, N2 gene, and RDRP gene, are relatively concordant. And you see a decay, as you would expect, with the little blips at 4 and 7 months, as we had noted earlier. If we try and look at this with time as a continuous variable on the x-axis, this is time on the x-axis and viral RNA concentration in copies per microliter on a log scale on the y-axis. You can see that we have the most shedding early on, but then we have people who continue to shed late. And one of the questions we have is, who sheds late and why? Is it something about their immune surveillance? Is it something about the strain with which they are infected? Is it something about hiding of these viruses in sanctuary spots within the body? Interestingly, when we compared the placebo control arm to the interferon lambda intervention arms, here you can see placebo control in blue, interferon lambda in interferon lambda. I'm just going to wait for the audio to be sorted. That sounds good. Okay, so the placebo control in blue, the interferon intervention in orange. Here we have baseline, 14 days and 28 days on the x-axis, the percent of positive subjects on the y-axis. Interestingly, you can actually find that fecal positivity is higher in the placebo control than it is in the lambda intervention arm. This is actually also consistent with the finding by Jagannathan et al. Reporting from the original trial that there was a higher baseline seropositivity in the control arm. And looking back at these data, one of the things that we hypothesize is that patients in the control arm may have been infected for a longer period of time by the time of enrollment. If we take a look at fecal shedding at baseline in the placebo control versus the interferon intervention arm, this is just a forest plot. Adjusted odds ratio is indicated on the x-axis, and you have the 3 different rows, baseline, 14 days, and 28 days. We see that there is a decreased or a higher odds of fecal shedding at baseline in the placebo control versus the interferon intervention. So once again, suggesting that while this was a randomized controlled trial, there probably was some slight imbalance in the arms. Now, I've shown you that we can measure fecal SARS-CoV-2 RNA well, and I've also shown you that fecal SARS-CoV-2 RNA continues to be shed long after symptoms are cleared and long after respiratory symptoms, respiratory shedding stops. But what is the consequence of this? Is this associated with GI symptoms? Is this associated with any other aspect of the disease? What we did was take a look at whether or not GI symptoms are associated with fecal shedding. And in this forest plot, we found what we believe to be one of the first reports of GI symptoms being associated with fecal shedding. Now, whether or not this is associated with a true GI infection or not, I can't say. But it's certainly interesting to note that we had a statistically significantly higher rate of nausea, vomiting, and abdominal pain in people who tested positive for fecal SARS-CoV-2 RNA versus those who tested negative. This is just a zoomed in version of that. So taking this all together, what have we learned? Well, we've learned that SARS-CoV-2 RNA is shed in the feces. We knew that before, but now we know this in a way that allows us to assess the dynamics of this shedding and to investigate the heterogeneity of shedding between individuals. We know that there are standardized methods for detecting SARS-CoV-2 RNA for feces that we've developed and that can be used more broadly. We know that fecal shedding continues after respiratory shedding stops. And while we still don't know whether or not there really is an infection of the gut by SARS-CoV-2, there is evidence for possible infection of GI tissue, at least using cell culture systems and organoids. And I've demonstrated some of the evidence that's in the literature that suggests that not only SARS-CoV-2 RNA, but also protein can be found in the gut long after respiratory symptoms and respiratory shedding has ceased. Now, whether or not the GI tract is truly infected remains unclear and an area of active investigation. We do think, however, that the understanding of the dynamics of fecal shedding will be highly informative for wastewater-based epidemiology efforts. And we've had the great fortune of being able to collaborate with Ellie Boehm and her group here in engineering to actually implement some of our learnings from fecal SARS-CoV-2 detection in their wastewater-based epidemiology studies. Some of the ongoing questions that we've asked with their group are, do all variants have the same amount of shedding? These are open questions that we anticipate future research will shed light on. So in terms of future directions, impact, and implications, as I mentioned, we hope that this will enable wastewater-based epidemiology efforts to understand population infection rates. We now have also developed methods to monitor viral infections in the GI tract, and we hope that this will be useful for future viral outbreaks, be they future SARS outbreaks or norovirus outbreaks. One of the things that was amazing to us when we embarked upon this investigation was that when we were looking for precedents for other monitoring of known viruses that infect the GI tract, there were very few papers that reported on this. And so we hope that this will form the baseline of future studies of all kinds of different GI tract infecting viruses. We also hope that this will form the baseline for exploring the role of possible prolonged antigen presence in the GI tract and the development of immunity and tolerance. And this is one of the things that we spend probably the most amount of time in our group talking about. What does it mean that we see antigen in the gut? Can we actually test for antigen in the feces? This is part of an active collaboration with Ben Pinsky's group. If there is prolonged antigen presence in the gut, how does that affect immune tolerance and the development of immunity? As we think about work in the allergy and immunology space, for example, there is a saying that antigens that our gut is exposed to induce tolerance. Antigens that our skin is induced to induce inflammatory responses. What if having infections in the gut by SARS-CoV-2 or similar leads to immune tolerance and an increased susceptibility to reinfection? There are many open questions and a lot of additional work to do, but at this point, I'd like to open it up to questions. I want to once again thank the entire interferon lambda team who did an amazing job of executing a world-class clinical trial during a very difficult time. I'd like to thank my entire lab, and I tried to call out the people who specifically contributed to this work. The Pinsky lab, who oversee clinical virology here and have been outstanding collaborators of ours from the beginning. The BAME lab in civil engineering, who has been doing wastewater-based epidemiology. They've been extremely helpful to us in helping us to develop rigorous controls and to put our work in context of greater population-based studies. I'd like to thank the Stanford APB. I don't think the APB gets thanks very often, but as we were all rushing to do SARS-CoV-2 research here and around the world, there are panels on biosafety that actually thought deeply and thoughtfully about how to keep our research subjects safe and how to keep our researchers safe. And I'm incredibly thankful to them for working way over time to ensure that we could all contribute to this important research and keep ourselves safe. Similarly, the Stanford IRB processed a huge number of proposals during this time and did so in an incredibly quick manner. We've also been collecting samples from other studies, including the FAVI study, so I'm thankful to the FAVI team, Christina Kong and the super team. Christina has overseen the Stanford wastewater-based epidemiology efforts, along with Ali, Ruth O'Hara, Bonnie Maldonado, and Catherine Blish, along with the entire group of researchers such as Press Jagannathan, Upi Singh, Julie Parsonet, Jason Andrews, Hector Bonilla, and Angela Rogers. So with that, I'll thank our funding sources, all of you, and I'm excited to hear your questions.
Thank you, Dr. Bhatt. That was excellent. And there are already a lot of questions coming up here. I also want to give an opportunity for our people, and there's a lot more people coming in. So we're already at triple the amount of people we had a month ago. So this is a great trend. So if anybody has any questions here, please raise your hand. We have a mic I can bring over to you. And I'll get started on a couple of the questions that are coming through here. So for those of you on Zoom, please do keep adding questions and of course vote the questions. Alan Ringgold asked, can another person catch COVID from a patient who is shedding?
Great question. So can another patient catch COVID from somebody who is shedding fecally? It hasn't been demonstrated yet. As I mentioned, there are very limited reports. I can count those reports on one hand of live SARS-CoV-2 being obtained from fecal samples. The one that I'm most familiar with is actually from an infant's gut, which is interesting, a kind of outgroup. We don't think of that patient population very often in terms of shedding. Right now, I would say that on balance, the evidence suggests that fecal oral transmission is very uncommon if it happens. Although I will say at the very beginning of the pandemic, when we heard about things like these cruise ship outbreaks, I think it led a lot of us to question whether or not there might be some fecal super shedders, because from an epidemiological perspective, man, that really looked like the type of norovirus outbreaks that we see in cruise ships. So, on balance, at this point now, 18 months plus into the pandemic, I think fecal oral shedding is certainly not the norm. And if it happens, is very, very unlikely. I wonder about whether or not there might be fecal super shedders. But I think at this point, we really don't have strong evidence for that yet.
Questions here. Linda Barman asked the question. She probably sees a lot of long COVID as well. So it's a great question. Could it be that persistence of COVID in the gut could be responsible for long COVID symptoms? Are there any studies on that?
Thank you very much for that question. This is actually something that Press Jagannath and one of our collaborators and Karen Jacobson are very, very interested in. And they're actually going to be leveraging our shedding information along with the extensive symptom collection data that they have from the Lambda trial. So I think that we will soon start to get the answers to these questions. Now, long COVID is still a relatively, I wouldn't say rare phenomenon, but given that fecal shedding, long-term fecal shedding is rare and long COVID happens only in a subset of patients, I do wonder if we're going to be underpowered with the Lambda study alone to be able to comment on that with any statistical significance. But once again, this is an opportunity to leverage other great trials and sample collections, such as the FAVI study that we have here, because a similar framework for data collection on symptoms was used for these studies.
To mix it up and go over to Dr. Berry here in the crowd.
Like that in the crowd. I mean, that was terrific as always. Two questions. One, in the prolonged spreaders, any of the comorbid diseases seen that might enhance spreading? And two, there's been some discussion about fecal aerosol transmission, the SARS-1 large transmission in the hotel. And there's been, I think, an episode in Boston, a super spreader events. Can you talk about fecal aerosol?
So I do think that the fecal aerosols are a very interesting concept. You know, you mentioned the conference that happened in Boston. It has been demonstrated actually that with SARS-1 and MERS, those viruses can be detected at the RNA level in the stool. And so I think there is a thought that there may be a subset of people who can shed live virus. You know, one of the questions I have is, well, maybe people who are on extensive proton pump inhibitors don't actually have killing of the virus in their gastric cavity, and those are the ones who end up having long-term infections. So I really do kind of wonder about this. And your other question was, remind me? Comorbid illnesses. So fecal shedding and comorbid illnesses is an extremely interesting area of investigation. I will say in this patient population enrolled in Lambda, people were relatively healthy, outpatients with mild to moderate COVID. So we weren't really well powered to investigate that. There was a really interesting piece that came out in the New England Journal of Medicine by John Lee at Brigham and Women's, where they looked at long-term respiratory shedder. And this is a person who is heavily immunocompromised. Not only did they find long-term respiratory shedding, but they actually found mutations of the virus. And so there has been this suggestion that immunocompromised hosts may end up being people who can house virus for a long time, and then those may be opportunities for the virus to evolve. And so I absolutely think that's an interesting question. I think it'll be very interesting to look at this in longitudinal samples from patients who have various levels of immunocompromise. I mean, one of the things I wonder about, although once again, we're not probably well powered to do this sort of analysis, is are the people who had long-term shedding not deeply immunocompromised, but in a very moderate or mild way, immunologically different, such that they have tolerance as opposed to inflammation and clearance of the virus? I mean, I will say this sets up an interesting opportunity to look at the intersection between microbes and the immune system, because we have this extensive experiment that has happened all over the world where everybody has been infected with or many people have been infected with a very similar virus, and we have a large spectrum of different responses. So I think there will be ongoing investigation on this topic.
I'm going to jump back to Zoom for a question, then I'll come to another live question here. Gil Chu asks, can stool monitoring of Stanford buildings correlate with fecal shedding with numbers of infected individuals?
So actually, this is a project that is underway. This is part of the super project that Christina Kong and Ali Bamer are leading. There has been some monitoring of wastewater effluent from certain buildings and the matching of those data to known COVID positivity rates of the occupants of that building. So this is actually, these are experiments that are underway. We can say that, you know, the rate of COVID positivity is still pretty low, but there does seem to be some correlation between COVID positive subjects in a building and wastewater effluent from the building. So it's actually fascinating, and we're very fortunate here at Stanford to actually have our own wastewater treatment facility. Many of you may not know we have Codiga, which is an on-campus wastewater treatment facility that not only manages our waste or much of our waste, but also serves as a research opportunity for many people here. So this has represented a really interesting lens, or offered an interesting lens into how to link information on individual infections with not big wastewater treatment facilities that are for an entire city, but for local treatments and outflow from buildings. So I never thought I would see the sewer maps for Stanford, but they're very interesting.
We'll move to a question from the audience live here in front of our residents. Thanks, man, for asking.
It's just amazing talk. You talked a lot about, I think, the intriguing hypothesis about immune tolerance with prolonged shedding. I was wondering if there's any data or any efforts in studying just in the general population reinfection rates with either just like prolonged GI symptoms or shedding, and whether that's the same variant or not, different variants. If it's the same variant, that may lend more evidence for that.
I think that this is an area that should be an area of future investigation. Now that we are seeing, unfortunately, reinfections, infections after vaccination, I think it's going to be a very interesting opportunity to capture individuals like this and actually answer questions such as, is it the same strain? Is it a different strain? How different does the strain have to be? Do we develop strain-specific immunity? And I think slowly but surely we'll be able to answer these questions. I do think what allows all of this is careful, consistent sample collection, careful and consistent clinical data collection, and standardized methods that allow us to compare between centers. So I hope that some of the things that we talked about today in terms of themes of how we collect samples will enable more comparable results, which will allow us to answer some of these questions that rely on us studying much bigger populations, right? Because when we're trying to figure out things about reinfection, we know that reinfection is a relatively rare event. So we need to study way more people to be able to answer those questions. I think those are outstanding questions, and I'm looking forward to seeing what we and others find.
A lot of great questions. Murray Solomon asks, how far away are we from a 23andMe microbiome evaluation for individual use?
This is the million-dollar question, I guess. This is something that many companies have gone after. I think many people are familiar with Ubiome, which was a company now defunct that actually tried to provide microbiome testing as a service. They started in what I would call the educational entertainment space or edutainment space. They moved into the clinical space. That's when they got in a little bit of trouble. There are some other companies out there that do offer that as a service, and also big research studies like the American Gut Microbiome Project, overseen by Rob Knight and colleagues at UC San Diego, where you can essentially pay to play, send in your sample, get some information back that compares your microbiome to others. I think at this point, the challenge is, what do you do with those data? I get emails on a probably weekly basis from people who say, hey, I sent my sample to fill in the blank company or fill in the blank study. I got this report. Now what do I do? And as somebody who spends most of her waking hours thinking about this, I think the challenge is we still don't know how to make sense of many aspects of a healthy person's microbiome. There's a huge amount of heterogeneity. We are very, very different from a microbiome perspective from one another compared to how different we are from a human genetic perspective from one another. And we also don't know a lot about the genomics or the microbiome composition of most individuals on Earth. So most of the microbiome research has focused on people in high-income countries. So I think one of the unintended consequences of these pay-for-play issues or pay-for-play options is that it heightens the gap between the haves and the have-nots, because we learn more and more and more about people who can afford to pay for these studies, and those who can't afford to pay for these studies are very poorly represented. So this has been an area of great interest for our lab. We try to work hard to try and bridge that equity gap in genomics research, not only from human genomics and disease genomics, but also for microbiome genomics. And we collaborate with the H3 Africa Genomics Consortium to try and address this. So, long story short, there are companies that enable you to do this. It's hard to know what to do with those data, but we are very much in progress. And I think we're very interested in figuring out, how can we measure something that actually matters to you? When can I do a test where I get a result and I can tell you what to do with that? I think that's when it becomes useful.
The questions keep coming. We'll try to get through as many as we can. Charles Wang asks, how has Delta changed the outcome/infection risks What are the risks to GI staff for colonoscopy and EGDs? What about seating of patient respiratory tract during these procedures?
I don't know is the short answer to this question, but I'm very interested to know the answer. When the Delta variant became prominent, we were very interested in as a lab in trying to understand if and how the GI tract might be infected with Delta variant. Could there be different amounts of shedding? Could there be more prolonged shedding? Are people with the Delta variant more likely to have live virus? Right now, we don't have the samples to be able to answer those questions yet, but I think this is an area that should be prioritized in terms of investigation. I also think that one of the questions we have to ask ourselves moving forward is what happens when we have the next variant and the next variant and the next variant. And I think that's when it becomes critical for us to find ways to distill down key learnings so that we not just describe that variant A is associated with this and variant B is associated with that, but we understand what the underlying biological processes are that might help us predict what happens when we get new variants. So I don't know the answer to this. I think these are very important questions, and I hope that sample collections that are now ongoing will help us answer, us and other labs answer this question in the months and year to come.
Excellent. Hector Bonilla asks, do you know if COVID has found pyre patches, any correlation of cytokine response, viral shedding in the stool? And finally, what is the role of persistent viral in the stool and long COVID?
So to the prior question, and Hector Bonilla was one of our amazing collaborators in the Lambda study, has thought deeply about all of these topics. In terms of pyre patches, there are no data to my knowledge that suggest that SARS-CoV-2 can infect pyre patches, but I will admit that I haven't actually looked at that question specifically. So I think it's interesting because obviously that's where a lot of the immunological training happens. So I think it would be very interesting to investigate that. Number two, in terms of, shedding and long COVID, we've touched on this a little bit before. I think this is going to require us to investigate data that we have. I'm actually going to speak with press later today, so hopefully we'll be launching some analyses of existing data and data that we can generate to try and answer that question.
Great. Georgina Hodgson asks, have you seen an increase in SARS-CoV-2 in patients who have celiac or Crohn's disease?
Good question. So for celiac and Crohn's disease patients, is there an increased risk of SARS-CoV-2? I don't know is the easy answer to that question. I don't believe there is an increased risk, but I haven't looked at this particularly. In terms of GI shedding and these patients, I think that would be interesting to investigate, along with patients who, for example, are on prolonged proton pump inhibitors or other kind of gastric acid suppression. So I think these are areas where we're probably starting to have enough patients to be able to answer those questions. Now, one of the challenges of these interventional trials that have collected samples very nicely is that, as with any interventional trial, there are often some exclusion criteria, and some of those exclusion criteria are, immunocompromised state, inflammatory bowel disease, other autoimmune disorders. And so I do think that some of these patients who might be more susceptible to SARS-CoV-2 in the gut may have been excluded from some of these big prospective trials. And so I think targeted sampling is going to be very important. But I know in a variety of different subspecialties, efforts have been made to collect samples from patients who are cared for by those subspecialties to answer some of these important, more focused questions.
Kenneth Weinberg asks, great talk, interesting study. Do you have any indication of whether there are specific gut cell types, regions, and stages of differentiation of infected GI cells? Are epithelium stem cells or stem cell niche cells infected? And would this be predicted to have a different consequence than infection of mature epithelium?
So right now, I think we don't know the specific answer to that question. I'm hoping that groups like Hans Cleaver's group and the Nussensweig group in New York are trying to address this with GI samples gut biopsy samples collected from patients. I think that this represents an outstanding opportunity to actually apply single cell sequencing technologies. And if I had, all the money in the world, that would actually be an experiment I would be excited to do. One of the things that we talked with our GI colleagues about was actually getting biopsies from patients with active SARS-CoV-2. Obviously, that puts practitioners at risk. And if there is not a clinical indication for doing those biopsies, it makes it a little bit complicated to achieve that objective. But I think that, some of the ways around this that have been very clever is early in the pandemic, people were still undergoing, screening colonoscopies, things like that. And a subset of those people ended up being SARS-CoV-2 positive, just by chance, because testing wasn't very prevalent. And that represented an opportunity to get gut biopsies from people who were healthy, otherwise healthy, who happened to have COVID. So moving forward, I think that's the experiment that I would do to try and figure out what cell types in the gut are susceptible to potential SARS-CoV-2 infection, collecting biopsies from patients with infections in a safe manner, and then doing single cell sequencing, or even, honestly, there are very, very cool in situ 2D approaches, such as, spatially resolved transcriptomics, multi-imaging, et cetera. So we really cool technologies that could help us answer those questions.
Excellent. Perhaps we'll do one more question and then we'll close it up. Edward Leibowitz is asking, do you include the liver and pancreas as potential sources of antigen shedding in addition to the GI tract?
This is a good question that we haven't thought about in great detail. Hector Bonilla has thought a lot about, whether or not the liver is infected. Obviously, areas of the liver, the pancreas, the gallbladder are contiguous with the GI tract because they're connected, obviously, by ducts. We haven't investigated this directly. I think it would be very interesting to consider.
It's right hitting 9 o'clock right now. I figure I wind it down. Thank you for all the amazing questions. There are a lot of great questions, both in person and especially a lot of them online as well. Sorry for the couple we didn't get to. Dr. Bhatt, thank you so much for being with us, and that was an amazing presentation. I just want to make a couple announcements. Next week, we'll have a lot of announcements of local updates regarding COVID. So please join early. We'll send some email updates about that. And I just want to say there was an email that went out yesterday about the Making Space program that Bob and Kathy really support. It's led by Rebecca Merrill, amazing personal coach. And one of the reasons I know Ami so well is because we were together in the Making Space program, and we're still close friends because of it. It's an amazing program and really a wonderful thing that the Department of Medicine leadership has for us. So thank you for that. And please do sign up for it for the faculty. Ami, thank you again. That was so wonderful. Thank you for being with us today and all the amazing work you do.
Thank you very much, everyone.